Okay, so previously we looked at the structure and the organization of the membranes. So with this, now we move on to the next part, which is regarding the process of transport, that how do membranes, they regulate the process of transport. So this is a figure here that shows different types of processes in the cell. So a cell is actually carrying out a variety of different types of transport processes. We're going to look at the details of some of these processes here. So for example, this is like an overview. There are carrier proteins which transport different types of substances in the cell. Say, for example, glucose is transported in the cell, and then glucose is metabolized through glycolysis to pyruvate, which is then further oxidized by the other oxidation processes, including the Krebs cycle, followed by electron transport chain, and so on. So thereby it produces energy in the cell. So the transport of glucose in the cell and then transport of the intermediates into the mitochondrial membrane, and then the redox reactions takes place, eventually would generate a gradient of protons in the intermembrane space. This gradient of protons is then used for synthesis of ATP. This process is also taking place on the membrane. So the protons, they would pass down the gradient into the matrix again, and as a result of which this would produce ATP. In the process, oxygen is required. So oxygen, being a non-polar small molecule, can diffuse into the cell and then can be transported to the mitochondria, and therefore will contribute towards production of energy. Then there are other different types of transport processes going on. For example, every cell requires to maintain a certain concentration of water molecules. So the transport of water is mediated by these special proteins, which are called as the aquaporins. Then there are other biosynthetic as well as catabolic processes going on in the cell. Likewise, for photosynthesis, plants would require carbon dioxide that is transported to the cells through diffusion, and then it would diffuse into the chloroplast, and eventually that can be used for photosynthesis. The metabolites, the intermediates produced by these metabolic reactions, are then transferred to other organelles, and in this manner they are then utilized for producing a wide range of different products. Amino acids, for example, can be imported into the cell by means of the carrier proteins, and also amino acids, they are produced by the degradation of existing proteins in the cell. So that way, different organelles of the cell, they are involved in the transport of different types of substances. For example, nucleotides being present in the cytoplasm of the cell are imported into the nucleus through the pores present in the nuclear membranes. Then the proteins which are required for the process of DNA replication, transcription, and other processes, those proteins are also imported from the cytoplasm into the cell, into the nucleus of the cell. There, this would result in producing RNA, and that RNA is then exported out through the nuclear pores into the cytoplasm, where the ribosomes can read that information and can make proteins. So, there are many different types of complex transport processes going on simultaneously inside the cell. So transport is not only taking place across the plasma membrane, but transport is taking place across all of the organelle membranes. So the membrane, one important function for the membrane is that it acts as a barrier and therefore can be used for regulating the process of transport. So the table here shows the three major important processes for um, for the transport. These include diffusion as well as active transport. Simple diffusion is a process in which molecules they move down their concentration gradient. So the region where the concentration of the molecules is high, they would start to diffuse to a region where their concentration is low. So this process does not require any additional input of energy, and this is called as simple diffusion. Typically, small molecules, non-polar molecules, they can be transported through the process of simple diffusion. For example, small non-polar or polar molecules, so these can be transported. Likewise, other non-polar molecules like the steroids or other different types of substances, for example, fat soluble vitamins or other substances which are non-polar, so they could be transported easily by the process of simple diffusion through the membrane. The other process is facilitated diffusion. Like simple diffusion, facilitated diffusion transports substances down the gradient, that is, from a region of a high concentration to a region of a low concentration. So, in this manner, the substances, the molecules, they could be transported. However, this process involves the engagement of proteins. There are specific type of proteins which are associated with the membrane. Most of them, these are integral membrane proteins. So these proteins are embedded in the membrane, and the proteins they bind to the different types of solute molecules on one side of the membrane, and then they transport them to the other side of the membrane. So this process is called as facilitated diffusion, because in this process, diffusion is facilitated by the proteins which are embedded in the membrane, and the proteins help in the transport of those substances, of those solute molecules down their concentration gradient. So, larger molecules, including sugars or other polar molecules like ions, so they could be transported by the process of facilitated diffusion down the concentration gradient. Since this process does not require um, is diffusion, so therefore it does not require any additional input of metabolic energy. Typically, that energy is provided by hydrolysis of ATP. Then is the active transport. Active transport process, typically they transfer the molecules from a region of low concentration to a region of high concentration. So this is against the concentration gradient. Active transport process is like an uphill task, so therefore it requires input of energy. Typically, that energy is provided by the hydrolysis of ATP. In some cases, other sources of energy, like a proton gradient, which is an electrochemical gradient, can also be used to power active transport process. So large molecules, which are typically polar or like ions, or when the substances have to go from again against the concentration gradient, so they are transported by the process of active transport. Simple diffusion is not regulated because this is simply occurring down the gradient. However, in facilitated diffusion, there are proteins which are involved, so therefore this process can be regulated. And likewise, active transport is also a regulated process. So this is a figure here that represents these various different types of transport processes in the cell. So this is representing a red blood cell. And in this red blood cell, different types of transport processes are described here. For example, the gases like the transfer of oxygen and carbon dioxide into the red blood cell or out of the red blood cell, these are simple diffusion processes. So these processes, they occur down the gradient. So when the red blood cell is present in the tissues of the body, the oxygen concentration in the so when the red blood cell is like coming from the lungs, so it is having high concentration of oxygen. So the concentration of oxygen in the tissues is low, therefore oxygen will diffuse out of the red blood cells into the tissues. On the other hand, the concentration of carbon dioxide would be more in the tissues, so therefore that carbon dioxide will diffuse into the red blood cells. When these red blood cells loaded with carbon dioxide, they reach the lungs. Now in the lungs, the concentration of carbon dioxide is low. So carbon dioxide will diffuse out of the RBCs. Whereas the alveoli of the lungs, they have a high concentration of oxygen. So oxygen will diffuse into the red blood cell. So this is representing the simple diffusion process. In addition, there are the other process is facilitated diffusion, and this process is mediated by different types of proteins. These proteins are called as the carrier or the permease proteins. So these proteins, they are important for the transport of substances down the gradient. Say, for example, this is a transporter for glucose. This is called as a glucose transporter. So glucose molecules can bind to this transport protein, to this carrier, and then the glucose molecules will be transported into the cell. In this manner, the process of movement of glucose from outside the cell to the inside of the cell will be facilitated by means of this carrier protein. Likewise, the anion channel, so that is important for the transport of bicarbonate and chloride ions. So uh, this is also an example of a transport protein. So this transport protein is important for performing the process of diffusion, but the protein helps in the process of diffusion. So therefore, the process becomes regulated. So both the glucose transporter and the chloride bicarbonate channel, so they both are examples of facilitated diffusion processes. This is sometimes also called as the anion channel or the anion exchange protein because it is going to exchange the two anions. This is important for the transport of carbon dioxide by the red blood cells. See that as carbon dioxide is transferred into the red blood cells, trans it is converted in form of carbonic acid, and carbonic acid dissociates to produce bicarbonate ions. So therefore, bicarbonate ions are negatively charged. So most of the carbon dioxide is actually transported in this form as bicarbonate ions. And when the carbon dioxide has to be given away from the red blood cell, so this bicarbonate will then be converted into carbon dioxide again. This is an enzyme catalyzed reaction. And when bicarbonate ions are pushed out of the cell, since these ions are negatively charged, so therefore the number of negative charges inside the cell will decrease. So therefore, to maintain electrical neutrality in the cell, so as one anion is exported out of the cell, which is bicarbonate in this case, another anion, which is chloride, will be imported into the cell. So in this manner, the total number of negative charges within the cell will remain the same. So bicarbonate exported and chloride imported, or vice versa. When bicarbonate is imported, chloride will be exported out of the cell. In this manner, the cell will be able to maintain electrical neutrality, meaning that the total number of ions in the cell will remain the same. So the glucose transporter and the anion exchange channel, they both are examples of the process of facilitated diffusion. This process is mediated by these carrier proteins. Then there are another type of proteins are called as the channel proteins. The carrier proteins, they have a binding site. So when the solute binds to them, they undergo a structural change, as a result of which they would transport the molecules to a particular direction. The channel proteins, on the other hand, they form a channel or a pore in the membrane, and molecules they can move across the membrane through these specialized channels. These channels uh, are actually made up of proteins, and they have a central cavity in them like a pore. So through these pores, the molecules can be transported across the membrane. One common example is aquaporin. Aquaporin is a channel protein or a pore forming protein which is important for the transport of water molecules across the membrane. And then is the active transport process. This process is dependent on the hydrolysis of ATP. So the energy obtained by the hydrolysis of ATP powers the transport process. For example, here it shows a sodium potassium ATPase. This is also called as a sodium potassium pump. So this is a transport protein found in the membrane. So the protein, it transports sodium ions out of the cell and potassium ions into the cell. This process is powered by hydrolysis of ATP. So in this manner, all these four different types of transport processes, including simple diffusion, facilitated diffusion by carrier proteins, facilitated diffusion by channel proteins, and active transport, are summarized in this figure. The movement of substances across a membrane is determined by the concentration gradient, or in case of ions or charged substances, it is determined by the electrochemical potential. So substances will migrate from a region of high to a low concentration by simple diffusion. So diffusion would move the solutes towards an equilibrium, which is that molecules will migrate from a region of high concentration to a region of low concentration. So this diagram is representing the transport of the gases in the red blood cell. So in the capillaries of the body tissues, so the concentration of oxygen is low, and the concentration of oxygen is low, and the concentration of carbon dioxide is more as compared to the red blood cells. So this is shown here that there is low oxygen concentration, so oxygen will diffuse. So these red blood cells are coming from the lungs, and they have a high concentration of oxygen in them, whereas the neighboring tissues have low oxygen concentration. So oxygen will diffuse out of the red blood cells, and carbon dioxide will be transported into the cell because the concentration of carbon dioxide is more in the neighboring tissue. So therefore, carbon dioxide will enter the red blood cell. Now, in the red blood cell, so there is this enzyme called carbonic anhydrase. This is a very important enzyme. The enzyme, it carries out the reversible conversion of carbon dioxide to carbonic acid. So this is a reversible process and can take place in both directions by the same enzyme, depending upon the concentration and other regulatory factors. So as carbon dioxide diffuses into the red blood cell, carbon dioxide is converted by carbonic anhydrase enzyme into carbonic acid. That carbonic acid will dissociate into bicarbonate ions, and this bicarbonate is then the red blood cells. They will then be through blood circulation will be transferred to the lungs. Now, in the lungs, the concentration of oxygen in the alveoli of the lungs is more. So oxygen will diffuse into the red blood cells, and the same enzyme, carbonic anhydrase, under the conditions of the uh, lung, so it will convert bicarbonate into carbon dioxide, and in this way this carbon dioxide will diffuse out because the concentration of carbon dioxide in the alveoli of the lungs is low. So therefore carbon dioxide will diffuse down the gradient. So as bicarbonate ions are imported into the red blood cell, so chloride ions will be moved out of the cell, and that it will be to regulate the total number of ions in the cell. That is important for, number one, regulating the electrical neutrality of the cell. That is, the total number of negative charges should be equal to the total number of positive charges. In this reaction, protons are shown, but protons are not the only positive ions. There are other positive ions like sodium, potassium, and others present in the red blood cell as well. So one function is to regulate the electrical neutrality. That is, the total number of negative ions should be equal to the total number of positive ions. So in this manner, there is a balance of um, bicarbonate ion concentration within the red blood cell. So this bicarbonate system also act as a buffering agent in the blood. So it is important to regulate the pH of the components of the compartments of the living systems. So in blood, the major buffering agent is the bicarbonate buffer system. So the bicarbonate buffer system is important for regulating the pH of the blood. The pH of blood is like close to neutral, slightly more than neutral. It's like 7.2 to like 7.4-ish, about that range. So the the major substance which is important for regulating the pH of blood is the bicarbonate buffering system. And red blood cells, they have this enzyme, carbonic anhydrase, which is important for regulating the conversion of bicarbonate to carbon dioxide, or vice versa, carbon dioxide to bicarbonate. In this manner, depending upon the concentration of carbon dioxide, so the enzyme can catalyze the reversible reaction in both directions. As a result of which, number one, this is important for the transport of carbon dioxide and excretion of carbon dioxide. Number two, this is going to regulate the concentration of bicarbonate ions and there in blood, and therefore it is important for regulating the pH of blood. Osmosis is also a diffusion based process, but in case of osmosis, this diffusion is taking place across the membrane. So and this is dependent on the water potential. So a region across the membrane which has more free water molecules will be having a greater water potential. So what osmosis is about, the movement of water molecules. So a region where there are free more free water molecules, they will migrate to a region where there are less free water molecules. So if the concentration of water molecules across the membrane is the same, so then the water potential will also be equal. So in this case, the solution is said to be isotonic. Iso means same, so we're having the same concentration. So if cells are present in an isotonic environment, the movement of water, since the concentration of free water on both sides of the membrane is the same, so the movement of water into the cell and out of the cell will be at equivalent rates. So the rates are comparable, so therefore there won't be any net movement of water across the membrane. However, when the cells are present in a hypertonic solution, so a hypertonic solution will have more solute concentration outside the cell. More solutes would mean less free water available. The interior of the cell will become hypotonic, meaning that it will have relatively less concentration of the solutes, hence more free water molecules available. So therefore, the water potential inside the cell will be more, and hence free water molecules will move out. The cell will undergo shrinking. This is a one typical example of this case is the the saline gargles. You see, so saline gargles help in relieving the symptoms of throat infection. Well, yeah, they mediate the effect by other ways as well, like you see, having warm water or like decreasing inflammation and so on. But one of the contributing factors is that saline gargles, since they have a high concentration of salt, so this is a hypertonic solution. So when this solution is present in the throat region, so it would essentially result in dehydrating the infectious bacterial cells which are present in the throat region, thereby um, inactivating them. So that will give sufficient time for the immune cells of the body to act against those pathogens present which are infecting the throat. So that is in case of a hypertonic solution. But when the cells are present in a hypotonic solution, so in a hypertonic solution, free water molecules will be more outside the cell. So the water potential will be more, so therefore free water molecules will migrate from the outside to the inside of the cell. And in this manner, the cell will start to accumulate water and will become bulged. Plant cells, they maintain turgid pressure by using this process. So the vacuole is hypertonic, therefore the water potential in the cytoplasm is more, so more water will be pushed inside the vacuole. And the vacuole, because of being more um hypertonic, so therefore the vacuole uh, will sort of imbibe more water. As a result, it will become swollen, will exert pressure on the walls of the cell, and the cell will become turgid. So this is important for preventing the plant cells from wilting and storing sufficient amount of water within the plant cell. So osmosis is a diffusion process across the membrane, and this is about the water potential, the movement of free water molecules across the membrane. So simple diffusion is important, regarding the small or the non-polar molecules. The rate of diffusion is proportional to the concentration gradient. So higher the gradient, rapid will be the rate of diffusion. The process also depends on the size, polarity, and the charge of the molecules. So molecules which are smaller in size, they are non-polar, and are uncharged, they will be able to diffuse rapidly as compared to molecules which have a larger size, are more polar or charged. So therefore, small hydrophobic non-polar substances, uncharged substances, they can easily pass through the membrane. Ions are, although they're small substances, but since they are charged, so they cannot freely move across the membrane. So therefore, special transport proteins are required to transport ions from one side of the cell to the other. Another common example showing polarity uh, and the effect of charge is is from the fact that when cells they acquire glucose, they uptake glucose. So most of the cells they convert glucose to glucose 6-phosphate. So glucose, although it's polar, but when converted to glucose 6-phosphate, now it will contain a phosphate group, which is a charge group on it. So therefore, this would increase the impermeability. So glucose will become impermeable to membrane upon phosphorylation. So in this manner, it does not diffuse away from the cell. So the process of facilitated diffusion is actually mediated by special type of proteins, and the process takes place down the gradient. So there are these transport proteins which bind and they transport the solute molecules to the other side of the membrane. This is a graphical representation which is showing this the process. So the red line, it shows the process of simple diffusion. It is dependent on the concentration gradient. However, the green line is representing the process of facilitated diffusion. So as you can see that the process of facilitated diffusion can transport the solute molecules more effectively and efficiently. And that's because of the reason that the protein, the transport proteins, they bind to the solute molecules and can transport them in a facilitated manner. So therefore, the process of facilitated diffusion will be more efficient as compared to simple diffusion. So the transport proteins, they fall into these two groups. One group of the transport protein, these are called as the carrier proteins, also known as transporter or permeases. So the carrier proteins, they have two conformations, two shapes. So in one shape, they bind to the molecules on one side, and then they undergo a structural change, and therefore they would release the molecules onto the other side of the membrane. Then there are the channel proteins. Channel proteins, they form pores in the membrane. And through these pores, substances can move across the membrane. So some of these pores, these proteins, these are called as porins because they form pores in the membranes, and therefore the porins, they can allow different types of substances to move through these channels or pores. So the channel proteins are selective in terms of the types of molecules they transport. So one group of porins are called as the aquaporins, and aquaporins are important for the movement of water molecules across the membrane. Then there are specialized uh, channels for the transport of different types of ions. These are called as the ion channels. Now, the carrier proteins, they can transport substances in different manners. One way is that the transport proteins, they can transport the molecules in such a manner that only one solute molecules is transported in a given direction. This is called as a uniport process. This is diagrammatically represented here in figure A. So you see S represents the solute molecule. So solute molecule binds to to the transport protein, and then it is transferred to the interior of the cell. So this is an example of a uniport process. So the transporter is called as a uniporter. Then there are other types of channel proteins which can transport molecules. Two types of solute molecules at a time. So there are two ways in which this is done. This is called as coupled transport. In coupled transport, two types of solute molecules are transported simultaneously. So for example, in the first case, you can see that solute A and B, SA, SB, they both are transported in the same direction. This is called as a symport. The other type of transport proteins, they will move the two solute molecules simultaneously, but in the opposite directions. So in this case, solute A is transferred into the cell and solute B is exported out of the cell. This is called as antiport. So the transport proteins, they can transfer the solute molecules either as a uniport, where only one solute molecule is transferred, or a coupled transport where two solute molecules are transported simultaneously. So in case of coupled transport, they could be transported in the same direction, known as a symport, or in the opposite direction, called as antiport. Glucose transporter is an example of a uniporter. So in purple color, you can see the glucose transporter is represented, and this transporter has actually two different conformations. The one conformation, which is called as T1, so the other conformation is called as T2 and is represented here. So you see that if you would compare T1 and T2, so in case of T1, the transporter is open to receive a glucose molecule from the outside. So glucose will bind here, and this would result, binding of glucose to the transporter would result in producing a change in the structure, as a result of which it will transition to this conformation called T2. I can see that in case of T2, the opening to the outside is now closed, whereas the region towards the interior of the cell is sort of like now open. So therefore, this glucose molecule can be transported into the cell. And as glucose is released from the transporter again will be converted into T1 conformation. So in this manner, glucose will be transported from outside the cell to the interior by means of the glucose transporter. So this is an example of facilitated diffusion. So the channel proteins, they actually form a hydrophilic transmembrane channel. And in this manner, the solute molecules, they can be transported across the membrane. So one type of these channel proteins are the ion channels. So ion channels, they are specific for the transport of different types of ions. For example, sodium, potassium, calcium, chloride, and so on. So these specific type of ions are transported by a specialized type of ion channels. Most of the ion channels are gated. That means the process of transport is regulated. So different types of stimuli can control the opening and closing of the gated channels. So the gated channels are like sort of like gates which can open or close under different conditions. And when the channel is open, substances can be transported. And when the gate is closed, transport cannot take place. So voltage gated channels, they open or close in response to the voltage across the membrane. Ligand-gated channels, they open in response to binding of special type of signaling molecules, which are the ligands for these channels. So when a certain ligand will bind to the channel, it will open or close. Then there are mechanosensitive channels, so which are based on the response to mechanical forces acting on the membrane. So the movement of ions across the membrane is important for various different processes. For example, the concentration of calcium, the transport of calcium regulates the concentration of calcium, and this is done by specific calcium channels. This is important for a variety of different things. For example, this is important for the movement of the muscles, like muscle contraction. This is important for cellular secretions. This is important for other processes as well. Then there are sodium potassium channels, and these are important for a variety of different things. One, regulating the osmolyte concentration in the cells and other body fluids, and then they're also important with respect to the generating the nerve impulse. So ion channels are important um, uh, for the transport of different types of ions. And a deficiency or like mutations, they can lead to producing an imbalance in the osmolyte concentration. One such example is a, a chloride channel. This chloride channel is called as the CFTR, which is cystic fibrosis transmembrane conductance regulator. This protein is a chloride channel, and it is important for the transport of chloride ions in the air passageways. So in case of a normal cell, so in case of the normal CFTR protein, so this is representing um, the cavity inside the air passageways, the lumen, and these are the cells on the producing the lining. And this is a layer of mucus secreted in the air passageways. So the CFTR protein is actually a chloride channel. It transports chloride ions into the mucus. Now, in order to maintain electrical neutrality, as chloride ions are pushed, sodium ions, as the counter ions, will also migrate into the mucus. Now, both of these ions, they are osmotically active. They interact and they are solvated by water molecules. So therefore, water molecules will also be transported into the mucus. So thereby, the mucus will have a certain suitable consistency and thickness. So, but in case of a mutation in the CFTR protein, so the transport of chloride ions does not take place properly. So since chloride ions do not migrate, are not transported, so therefore this will result in uh, insufficient uptake of sodium and therefore insufficient uptake of water molecules. So the mucus will not be hydrated. The mucus will become dehydrated and it will become sticky and hard, and this would eventually result in breathing disorders. So cystic fibrosis is a condition in which the mucus, it becomes very sticky, hard, and therefore it leads to breathing problems in that patient. So this is caused because of a mutation in the CFTR uh, protein. So this is a diagrammatic representation showing the CFTR protein in purple color, and this is a nucleotide binding domain shown here. So and close to this spot is the region where the mutation of an amino acid takes place, as a result of which the channel will not be able to transport the chloride ions out into the mucus. So, therefore, the channel proteins, ion channels, are important for the transport of different types of ions across the membrane. Then there are porins. Like porins are important for they form pores in the membrane, and they are important for the transport of different types of substances. Like aquaporins, they transport water. So porins are important for the transport of many different types of substances. For example, mutation in bacterial porins can lead to antibiotic resistance because when these porins will be mutated, so that will decrease the efficacy of the uptake of the antibiotic. So therefore, the bacteria would become resistant to the antibiotic. Aquaporins are important for the transport of water, and this is important for a variety of different things. For example, like in the kidneys, so when they reabsorb water, so that is done by a special type of aquaporins. So this is a figure representing the typical structure of porins. So in panel A, you can see this is the side view of the porin protein. This part is embedded in the membrane. And as you can see, it is largely made up of sheets. The sheets are represented by means of these arrows. So you can see that the sheets are forming a barrel-like structure. And this is panel B shows the view from the top. So you can see that in the inside, this is having a cavity. So through this cavity, through this channel, the solute molecules, they can move across the membrane. So this is a barrel-like structure made up of beta sheets. So sometimes this is also called as a beta barrel. Panel C shows aquaporin. So in aquaporin, you can see that there are 4 cavities inside through which water molecules can be transported. Active transport processes are mediated against the concentration gradient. So there are various important functions. So for example, um, this is important for the uptake of the nutrients from the environment. Secondly, it is important for processes like secretions or waste materials to be removed from the cell. And then it is important for maintaining a suitable concentration of substances required for different types of processes. So the active transport is mediated by these specialized proteins, which are called as the sort of like a pumps. And this process is a unidirectional process. So active transport process can be done like in 2 manners. So the first part is the direct active transport process. So as you can see here in the figure that this is a protein embedded in the membrane, which hydrolyzes ATP and then is going to pump the solute particles here. So the substance which is going to be transported, this is against the concentration gradient. So therefore, it requires input of energy by ATP hydrolysis. The substance being transported here are the protons. So in this manner, this is a process in which energy is obtained directly by the hydrolysis of ATP. So this process is direct active transport. In case of indirect active transport, it's not ATP which is used to power the process, rather it is typically a concentration gradient of the protons built up by hydrolysis of ATP, which can be used to power the transport of a substance. So for example, in the second part, you see this protein on the right side that transport the solute S across the membrane. And again, this is against the gradient. The concentration of S is more inside the cell. So this process is not powered by ATP itself. The process is actually powered by the proton gradient. So the high concentration of protons, as the protons move down the gradient, so the energy from the electrochemical gradient of protons is used to transport the substance S. This is an example of indirect active transport because the energy is provided by the proton gradient, not by ATP hydrolysis itself. So how was the proton gradient established? Well, that was because of the ATP hydrolysis. So therefore, this is an indirect active transport process. So in direct active transport, ATP hydrolysis is involved. In case of indirect uh, active transport, ATP hydrolysis does not occur directly. It's not coupled to the process. Rather, typically, it is a, an electrochemical gradient of the protons. There are different types of ATPases which are involved in the process of transport. These are called as P-type, V-type, F-type, and ABC type ATPases. They are different in terms of their structure, cellular localization, and their functions. Now we look at one important example of sodium potassium pump, which is an example of a direct active transport process. So the pump is actually going to push sodium ions outside the cell and pump potassium ions into the cell. So sodium goes out and potassium comes in. So this pump is made up of these two types of subunits, the alpha and the beta subunits. The pump can exist in two states. In one state, it is open on the inside, so sodium ions can bind, and then in the other state, it will be open from the outside, so as sodium ions can go out and potassium can come in. So cells, they maintain a high concentration of sodium ions outside the cell and a high concentration of potassium inside the cell by using this sodium potassium pump. This is how the process takes place. So the pump is present in this conformation. So 3 sodium ions bind here, and this process is then facilitated by the hydrolysis of ATP. So by hydrolysis of ATP, phosphate is released, and that phosphate will be covalently attached to the pump. This will change the structure, the conformation of the pump. So it will change from this even state to the E2 state, where the sodium, so now you can see that the pump is like closed from the inside and open to the outside. So these sodium ions will then be pushed out of the cell. Now this is open on the outside, so 2 potassium ions will bind here, and upon binding of potassium, the conformation changes, as a result of which the pump will go back to the even state, where the potassium ions will be transferred to the interior of the cell, and now the pump will be ready for another round of transport. So 3 sodium goes into the cell, 2 potassium goes out of the cell, and the process is powered by ATP hydrolysis. So ATP hydrolysis would result in producing phosphorylation of the channel, as a result of which the pump will become phosphorylated, and hence it will open on the outside, push the sodium out, takes the potassium, and then transport them into the cell. So that's how the sodium potassium ATPase works. So another important example is the indirect transport, and that is depicted by the symport of glucose. So this is a glucose transporter here. So two sodium ions bind here, and binding of the sodium ions facilitate the binding of glucose as well. So in this manner, glucose binds to the to this symporter, and now the symporter undergoes a structural change, opens on the inside. So therefore, sodium and glucose are then released into the cell. These sodium ions are then pushed through the sodium potassium ATPase outside of the cell, as a result of which the glucose molecule will be released into the cell. Hence, the process of symport will take place. So in this process, the energy which is used to transport glucose is provided by the movement of the sodium ions. And how did the sodium ions they were transported out of the cell? Well, that was because of the active transport process, the direct active transport, where sodium potassium pump has pushed the sodium ions out, and then sodium along with glucose is symported into the interior of the cell. So the process is powered by the gradient of sodium ions. In this manner, the two important types of uh, active transport processes are correlated to each other, direct and indirect.